There you go.
Welcome back to the STEMinars series hosted by the American Chemical Society student chapter at California State University, San Marcos. My name is Amanda Melanes, and I am an officer at the ACS student chapter at San Marcos, and I'm going to be introducing the event today. So thank you for joining us today. This is the third and final STEMinar in the series. Today we do have Patrick and Tiffany presenting. If this is your first time joining us, we just want to introduce the ACS officers at San Marcos who helped organize this event. And this is also in collaboration with Princeton University. So I'm going to hand it over to Vin, who's going to talk a little bit about their program.
Yeah, my name is Vin Tan, and I'm the events coordinator for the Princeton University Chemical Society. And on behalf of everyone at PUCS, I'd like to sincerely thank the student ACS chapter at Cal State San Marcos for this wonderful collaboration. And we'd also thank Patrick and Tiffany for speaking today, and we sincerely hope you enjoyed today's seminar. Back to you, Amanda.
Thanks, Vin. So just a little bit about the agenda today. Patrick is going to present first, followed by questions, and then Emily is going to introduce Tiffany before she presents, and her presentation will be followed by 10 minutes for questions. And then after, the live recording and the YouTube session will close, and there'll be a private Q&A discussion over Zoom. So if you don't know how you ask questions, if you're in the Zoom audience, you'll raise your hand using the participants window and a moderator will call on you, or you can type your questions in the Zoom chat. If you are joining us from the YouTube audience, you can email your questions to steminar.questions@gmail.com. And finally, if you'd like to connect with us, you can follow us on Instagram and Twitter at CSUSM_ACS, or you can email us at csusm.acs.club@gmail.com. Before I introduce Patrick, I'd just like to say a quick thank you to everyone who is watching today or is watching later on. If you missed the first or second seminar, you can also watch them on YouTube. So with that, I'd like to introduce Patrick. He is a fourth-year graduate at the Macmillan Lab in Princeton, where he is researching the direct aerylation of strong aliphatic CH bonds. Before going to Princeton, he got his undergraduate degree in chemistry from Wesleyan University in Connecticut, where he worked with Albert Fry and then Michael Kelter. After graduating, he spent 3 years working in industry at Novartis. So everyone help me in welcoming Patrick to the live podium.
Thank you so much for the very kind introduction, and it's really exciting to get the chance to talk to you all about a little bit of my path to graduate school and some of the work that I've done since starting here. So to begin, having watched several of these seminars now, they usually begin with something like this, where you show a beautiful map of the US that you can show how you sort of go from one side of the country to the other side of the country and back and work with all of these illustrious people. And I was going to do that until I put all the dots on the map and realized that it just looked sort of silly. As you can tell, they're maybe all within 200 miles of each other. So I thought it might be a little bit more helpful to go with a slightly more zoomed in map. So to begin, I was born on Long Island, which is just east of New York City. Which is a very nice place to grow up, in that there are lots of beautiful beaches that were only a few miles from where I was born, and that we're known for high cultures such as bagels and pizza and breakfast sandwiches. And when I was growing up, I had fairly diverse interests, and one of my first interests was baseball as a fairly young child, but I realized, being below average for my Little League team, that I was probably not ever really going to make it as a professional baseball player, given how many Little League teams I recognized there probably were, and that even the people who were really good on them probably weren't going to make it. When I was in middle school and high school, I became very interested in music. I played violin and then switched to playing guitar like many 14-year-olds. And I realized that as much fun as I was having playing guitar, there were many people who were much better at it than me. And even they sometimes struggled. So it seemed to me that that might not also be a particularly good long-term plan. So from that, I had fairly diverse interests in classes in high school, but I really enjoyed particularly a lot of my science classes. I had a really excellent chemistry teacher, and it probably helped that he was also in a very good band that I went to see a few times. So it seemed based on that like, oh, my chemistry teacher's band could be pretty good and the classes are fun. Maybe I'll study something like that. So I applied to a sort of broad range of different schools when I was applying to undergraduate. I actually ended up only applying to Wesleyan because there was no supplement and a friend told me I would like it and I sort of wanted one or two more to add to the list. And I ended up actually going there after visiting in part because of the really cool interactions I had with the chemistry department. And one thing that I had not really understood at all until probably around the time I was graduating actually, is that the school I ended up deciding to go to, which is this small, primarily undergraduate institution, actually had a remarkable tradition of organic chemistry research. And the reason why turns out to be that this quite famous chemist who actually won the National Medal of Sciences in the 1980s, named Max Tischler, for reasons that are still unclear to me, decided that when he was going to have to retire from Merck, because they had forced retirement at that time, that despite all of the various offers he would have had to work as an academic in various more famous institutions, he decided that he was going to join the chemistry department at Wesleyan University. And because of that, we actually had a really, really great group of professors who were hired from around that time, who are now relatively old professors. And it really started this great tradition of organic chemistry research at Wesleyan. So at the end of my first year, I was talking to a friend who sort of understood how science and undergraduate worked probably better than I did. And she realized she was talking about how she was going to join a research group because she was interested in doing science research. And I thought that probably sounded like an important thing to do as well. So I should probably do this too. And I ended up speaking to my really great undergraduate advisor, Albert Fry, who had convinced me at the beginning of that year to take his intro organic chemistry class. And after sending out emails to a range of professors, I ended up telling him that I really wanted to work with him over the summer. And he gave me an interesting project to do that I didn't really understand any of the details of at the time when I was first doing it. But looking back over my notes, I can see that what I was looking at was the reduction potentials of a range of aryl substituted aldehydes using a technique called cyclic voltammetry, which I didn't really understand at all at the time, but was still a really cool opportunity to sort of get my hands wet and get a chance to do some work in an actual lab on an actual research problem. So after that, I got an email after probably had been in Professor Fry's lab for 2 or 3 months that he recognized that he was probably going to retire before I would graduate and thought it would be better for me to find an advisor who would be there for the entire duration of my undergraduate. As a side note, it turned out later that he actually did not end up retiring and was maybe just trying to get rid of me. But regardless, I ended up switching to a more synthetically oriented group in the lab of Professor Michael Coulter, where we used these natural product-derived catalyst scaffolds. In this case, what I'm showing here is the structure of quinine as the way to develop methods for the asymmetric synthesis of important organic molecules. And looking back over the work that I did there, I really enjoyed looking at sort of these two specific reactions. Because if you were to look at it from the perspective of a graduate student in chemistry or someone reading the literature, you would look at these and see that these are both effectively useless. So these reactions does not proceed in particularly good yield. The enantiomer enrichment is very, very poor. This would not be publishable at all. And while this one still worked in reasonably good yield, I got stuck at sort of the classic 70% EE that if you talk to people who work in this field is a problem that you run into fairly often. But what I find so cool about this is that even though these results are not particularly helpful for anything, the work that I did in Professor Coulter's lab was very, very instrumental in where I've ended up coming now. It was what enabled me to do the work that I did after graduating in industry and what set me up for my career in graduate school. So I think this is a really great illustration of the importance of undergraduate research, and that even if it turns out that everything you work on doesn't work, which happens all of the time in scientific research, and perhaps particularly when you're an undergraduate, you can still learn a ton from it, and it can still be a very, very important stepping stone for your career. So I spent 2 of my summers in undergraduate working in Professor Coulter's lab, as well as some time during the semester. But I was also fortunate enough to get to do a DAAD RISE fellowship in Germany. The DAAD is the German Academic Exchange Service, and they have this great program where they can take about, I think, 300 scientists from the English-speaking world every year and pair them up with some academic group in Germany where you'll work there for the summer. And it also comes with, I think, a 2-week language class in Berlin, which was a lot of fun. And working in Germany, I got to work with a great assistant professor at the time, Anna Staubitz, who's now an associate professor at University of Bremen. And this was the first chance I got to think a little bit about photochemistry, which is a field that I now spend a lot of time thinking about in the context of these what are called spiropyran molecular switches. And these struck me as very, very cool because if you look at the structure on the left, you have this insulating structure, but you can actually irradiate it with a specific wavelength of light. In this case, it would be near UV light to convert it into this conducting polymer. And furthermore, you can switch this conducting polymer back into this insulating polymer by irradiating it with a different wavelength of light. And this was really cool to us because you could imagine basically putting this as a switch in the middle of a long conducting polymer. And it would be an interesting way to think about making these sort of interesting light-dependent photo switches. But even more importantly than the research I got to do there, it was a really cool opportunity to live somewhere different for a year or for a few months and actually get paid for it. So the city that I was working in at the time is a city called Kiel, which is on the coast of the Baltic. And it's just a really beautiful, very classic European student city, essentially, where it's about, I think, 200,000. And basically everyone bikes everywhere. And it's a short bike ride from the beach. And on like the few days where you actually get good weather, everyone just hangs out at the beach all day. So it was a really cool opportunity. And I think this is a program that is maybe not as well advertised as it should be. I just found it on the ACS website, and I don't actually even know all that many people who are familiar with it. But at least according to the program website, though the events of the last few weeks might call us into question, they do claim that they expect that the program will resume as planned in 2021. So if any of you are all interested in this program, I'd be happy to answer questions about it or provide further information. But it does at least look like they're planning on having it next year, though we'll see how the next few months go. So at that point, I'd gotten a lot of opportunities to do really interesting scientific research while I was an undergraduate. And at the beginning of my senior year, after I had at that point done 2 summers of working with Professor Coulter and 1 summer of working with Professor Staubitz, I was trying to figure out whether or not I wanted to go to graduate school. So one of the things that I found sort of intimidating about applying to graduate school was that when I thought about what my career trajectory might be, if I applied straight to graduate school, I would go and probably work in someone's lab for 5 or so years. And then the obvious subsequent path would be to do something like a postdoc for another 2 to 3 years after that. And then maybe from that, I would go straight into applying to some kind of academic position would be the traditional route. And that sounded like something that was interesting to me, but it also seemed maybe more specific than what I felt comfortable with committing to at that time. I knew that I really, really liked chemistry, but I also was someone, as I mentioned before, who has always had fairly broad interests. I wasn't sure I really liked it that much. But then this left me with an even more confusing question, which was, what do I do if I don't apply to graduate school? When you're in high school, it's very clear that your next step would be to apply to undergraduate. And if you're doing well and very interested in your field in undergraduate, it's a fairly clear next step to apply to graduate school. But once you decide to sort of veer from that path, the options become in many ways overwhelming and sometimes like sort of much less limited and much less clear. And I was furthermore not even certain exactly what kinds of jobs I would even be qualified for as someone who just held a bachelor's degree in science. It seemed to me, based on my limited understanding, that a lot of science was done by people who already have doctoral degrees. So in some ways, my decision was probably made for me by the fact that I went back and forth about this for a while, and it was starting to come time to submit applications. But in the fall of my senior year, I decided I was going to take a year off to work somewhere, and the plan at that time was to apply to graduate school after that year. So before I move on to the rest of my talk, I will give one piece of advice that I think if you all remember one thing from this talk, it should be this, which is that if you are considering taking time off before applying to graduate school, which is something that worked out very well for me, you should try and take all of the required exams that you will need for your graduate admissions while you're still in school. So it's something where it's much harder to get yourself studying for long exams when you're not used to that sort of lifestyle. And speaking to one of my good friends here who took the same path I did, where he worked for a few years before going to graduate school, he did not take his exams while he was still an undergrad. And that meant he spent like a lot of his evenings while he was working, prepping, and had to take a day off of work to go take his GRE. So I do think it's something while you're sort of in student mode and very good at studying for and taking exams, while it might end up feeling like a waste if you end up not going to graduate school because the exams are very expensive, it's something where if you're serious about applying, this would be a way to give yourself a substantial step in the right direction while you're still an undergrad. So with that in mind, I fortunately had a fair amount of time after I had decided not to go directly to graduate school to think a little bit about what I wanted to do and apply to jobs, particularly in the spring of my senior year. And I ended up getting my first offer from a small company called New England Discovery Partners, just about 15 miles south of where I went to undergraduate. And there I actually found the position through my advisor. So while I was also applying to many things online, I actually found this one just because this was a company that my advisor was a consultant for. And I learned, and I actually did not get the offer until the day before I was graduating. So I essentially had no idea what I was planning on doing until the day before. And once I received the offer, I found out I was going to be employee number 5 and would be given a 6-month contract to do science there. And they had a very, very simple business model that was at least easy to explain to my parents what I would be doing, where companies would send them synthetic targets that they needed. These would often be things like food and beverage companies that would maybe need to do a safety test on some sort of additive, but don't really have a dedicated organic chemistry group that might work on it, or a range of other sort of not often pharmaceutical companies, but other fields that would require organic synthesis. And then once they sent us these targets, it would be our job to make them. And I actually learned a remarkable amount at this job for, as I'll get into shortly, how little time I spent working there. And the main thing was that you can do incredibly good science with very little infrastructure. So this lab, I think, had one very old GCMS, one HPLC with a single column, and TLC plates, and they actually did have one automated column machine, which was used essentially continuously. But with that equipment, they were able to make complex molecules very quickly with beautiful routes. And it really taught me all that you can conclude from very classical techniques, like looking at a broad range of TLC stains. And a lot of those skills have stuck with me till today. And the other thing that I found really interesting about it is applying to jobs in science as an undergraduate. I was a little bit concerned about whether or not I would even be qualified for anything like this. But I actually found that as soon as I started at the CRO, which is, I may have forgotten to define this before, but it's a contract research organization, that the day-to-day work I did there was remarkably similar to the work I did day-to-day as an undergraduate. So the way I would be making starting materials and then testing reactions while developing my asymmetric catalytic methods with Professor Coulter was actually quite similar to the way I would be trying different routes to different targets that we were asked to synthesize as part of New England Discovery Partners. But one thing that I had not anticipated at all is, as I mentioned before, I applied to quite a lot of jobs. And less than 2 weeks after starting my position at New England Discovery Partners, which was essentially my first real job, I received essentially an order of magnitude better offer from a different company. So at that point, I actually quit at New England Discovery Partners, told them I was very sorry, but I'd received an offer that I was not expecting to receive that was substantially better for my career. And since I had some time and I felt bad about leaving so quickly, I actually gave them 3 weeks notice. So it meant that at my first job, I actually worked there longer after quitting than before, but they were very understanding. And this led me on to the next chapter of my time before starting graduate school, which was moving from the New Haven area up to Cambridge, Massachusetts to work at Novartis, which is a very large pharmaceutical company where their headquarters of scientific research were at Cambridge, where I would be working. And I actually got this Novartis job just by applying to, I think, an Indeed.com link for a posting for a medicinal and synthetic chemist. I ended up really liking the job and working there for a total of just under 3 years. And the way the job worked could be described something like this. So I imagine you guys have seen slides that look something like this in the past, but the drug discovery process generally begins with very broadly looking at a large range of organic structures to see which structures might bind to your potential drug target well, or might have the kinds of properties that might make them useful oral drugs. Then you'll move on to a step where you take a smaller number of compounds and characterize them in greater detail to figure out what might be interesting molecules to test in clinical trials. Then from there, you go down to definitely single digits, usually only 1 or 2 drugs that you'll actually conduct clinical trials on. And then these will, if all goes well, make their way to FDA review and hopefully become an approved drug. And as a medicinal chemist, which is what I worked as, you spend most of your time thinking about this first step of the process, where you do everything from interpret the results of what's called a high throughput screen, which is testing literally often millions of molecules against a potential target and seeing if any known structures inhibit it in a way that you would like. Then converting those what are called hits, so those molecules from your high throughput screen that showed activity that looked interesting, into molecules that bind to your target better and show interesting promise as a potential drug, and then the further optimization of that structure to get to what will eventually be the compound you'll test in clinical trials. And the way this works in a day-to-day sense is something like this, where you have this iterative process of designing new molecules, figuring out how you'll synthesize them, making them, testing those molecules that you've made, and then looking at those results and using those results to design new structures. And what this enables you to do is define what is described as structure-activity relationships, or SAR. And this is really most of what you'll spend your time doing as a medicinal chemist. You'll be looking at both the relationships between the structures you've made previously and the activity as far as something that maybe inhibits your target well or has properties that seem promising for becoming an oral drug. And then you'll decide, based on this data, what new compounds should we consider making. And then you will plan synthetic routes and execute those routes in the lab. So this is basically what I spent every day doing for 2.5 years, and it was a very rewarding job, which was in many ways a lot of fun because of how interdisciplinary it is. So every day along all the steps of this process, when you're looking at sort of the testing of your molecules, you're interacting with a lot of biochemists. When you're thinking in more detail about the target and what could be maybe potential benefits or disadvantages of targeting that specific protein, this involves lots of discussion with people who think very seriously about biology and medicine. And then you also spend a lot of time thinking about organic synthesis and what is the most efficient way to make molecules in the lab. So before I move on to talk about the work that I've done in my graduate school, I think it's worth going into some detail about one of the projects I got the chance to work on as a medicinal chemist as well. So you guys can see a little bit about what that work looks like. So before I got to Novartis, they were interested in this protein shown here called SHP2, which it was known is upregulated in a large number of cancers. And that this came from Novartis thinking very broadly about what are the kinds of proteins that are either overactivated in some sort of way in cancers. And one thing that was interesting about this molecule is that although its active site, which is basically this little spot here, was shown to be extremely challenging to target because it is a phosphatase, which means it is highly charged, and these sorts of highly charged molecules tend to make very poor drugs. What was known is that it actually only enters the conformation in which this active site is accessible after binding to an exogenous molecule. So what they thought was there might be some way to stabilize this closed conformation, which would then allow you to inhibit SHP2 without needing a molecule that binds tightly to the sort of highly charged pocket. And this was a key scientific question that they worked on for several years before I joined the program. And what they actually found was after several years of optimization, they had this beautiful structure here, which was able to actually successfully stabilize this closed conformation, which prevented the activity of SHP2 with an IC50, which is the concentration of this compound at which the activity of SHP2 is reduced by 50% of 71 nanomolar, which means it's a very, very potent binder. But of course, when developing a drug, you don't stop there. So it's one thing to make a compound that binds tightly to your protein. It's very different to have one that has the sort of metabolic and permeability and stability properties that will enable it to be a useful drug. So while I was at Novartis, I got the chance to work on these very beautiful series of compounds. And fortunately, actually, just before this talk, a few weeks ago, this final compound was published in the Journal of Medicinal Chemistry. And this is actually a compound that is now in human clinical trials. So there are literally people who have cancers that cannot be currently treated who are being given a compound that is from a project that I worked on, which really, to me, helped sort of clearly illustrate the importance of the things that the seemingly esoteric academic things you learn about organic chemistry being able to really impact people's lives. So after working at Novartis, I was able to conclude two sort of clear things about chemistry that I feel like many people probably could have figured out without having taken this 3-year detour. But I knew from the experience I had developing molecules that ended up even making it all the way into the clinic is that chemistry is incredibly important, that advances in chemistry can literally dramatically improve people's day-to-day lives. And I also learned from the amount I enjoyed my work at Novartis that chemistry is incredibly fun. So based on that, I decided that although it is entirely possible to have a great career as just a bachelor's level scientist in chemistry, there's definitely a ceiling that you eventually hit in several ways. So it seemed to me that if I wanted to stick with it, it probably made sense to get sort of further academic credentials. And I also really enjoyed the academic aspect of my work, even at Novartis, where I enjoyed thinking about new kinds of reactivity that might enable you to access new compounds more quickly, or enjoyed also thinking about the kind of physical organic parameters that might affect things like binding to a protein in the context of protein inhibition for developing a new drug. So with that, I decided to apply to graduate school and make my way back to academia. And after visiting a number of schools and thinking quite hard about what might be interesting, I met this very charming Scottish man named Dave McMillan, who told me about the really cool chemistry his group has done, where they could shine light at reactions and get whole new kinds of reactivity than what they'd observed previously. And what this fundamentally came down to was that they had a way where if you have catalysts that absorb light, it could enable you to selectively generate extremely high energy species. So with that as my essentially understanding of how photoredox catalysis works, I decided that I would move from working at Novartis as a medicinal chemist back to being a student again at Princeton, New Jersey, which is shown here and is basically the exact same distance from New York as where I grew up, but on the other side. So with that, I'll move on to talk a little bit about the work I got the chance to do during my time in Princeton so far. So to begin, I imagine you all have seen this in some of the other seminars that have been presented, but palladium-catalyzed cross-coupling and metal-catalyzed cross-coupling in general had had a dramatic effect on organic synthesis over the last 30 to 40 years. With, for example, all of these pharmaceuticals being made in these key sites via a palladium-catalyzed transformation. And this has been so important that actually, as of 2010, the Nobel Prize was awarded to Richard Heck, Iichi Negishi, and Akira Suzuki for the development of these kinds of reactions. And the way they generally work is through the reaction of some sort of nucleophile, which are these class of molecules here, which tend to be these organometallic reagents, and some sort of electrophile, which is like the aryl halide shown here. In the presence of palladium can form these cross-coupled products. And while these electrophiles themselves are actually quite convenient, these sort of aryl halides or alkyl halides are very stable and relatively easy to access. These nucleophiles are a little bit less convenient in that they generally take several steps to prepare, are sometimes water or moisture sensitive, and sometimes have other unpleasant properties such as these alkyl tins here. So these sorts of traditional functional handles that are used in these kinds of cross couplings have been incredibly helpful for enabling people to make organic molecules in a sort of streamlined, highly modular fashion that's very important for the development of or the synthesis of large numbers of analogs for the things like drug discovery. But it would actually be much more interesting if we could use the kinds of functional groups that are actually already present in lots of organic molecules. So instead of having to convert something to a boronic acid and then do your key step, what if you could just begin with an alcohol that was present in a molecule that you already had to begin with? This would be generally quite helpful because it would enable you to reduce the length of your syntheses, because instead of needing to spend several steps pre-functionalizing each of your components, you could just begin from the kinds of abundant and generally stable functional groups that are already present in organic molecules. But if you look at all these abundant functional groups, one thing that you'll notice is that actually all of these molecules have another functional group that's even much more abundant, that's actually so abundant that it would be very inconvenient to draw them on organic molecules, and that is namely CH bonds. So one of the questions I was really interested in when I was beginning my time at graduate school is whether we could find ways to use things like CH bonds in the same way we think about using a Grignard reagent or an organotin in a highly modular sense as a point for diversifying organic structures. The way we thought about doing this is through this elementary step here called hydrogen atom transfer, where an open shell species represented by this X dot here removes a proton and an electron from a organic molecule to afford a reactive alkyl radical. This has been done in the past with a range of different species, most notably with things like halide radicals in traditional processes for the chlorination or bromination of hydrocarbons. And one thing that was really attractive about an approach like this is that once you've developed a method to access this free radical, in principle, you could use a broad range of subsequent steps to functionalize it. So you could imagine once you have a method for generating this, you could then put it in the presence of a brominating reagent or a what's called here a Manishi acceptor, or what's called here a Michael acceptor to generate a range of different products from it. In the same way that, as Tiffany will talk about later, photoredox catalysis has enabled similar approaches using the carboxylic acids or alcohols I showed on the previous slide. And what this does is it does enable a really modular functionalization, where you could imagine if you have different sorts of X dot, you could abstract CH bonds from different positions to generate different alkyl radicals, and then each of these can be divergently functionalized to a range of different interesting products. This was used by the research group that I'm working in, the Macmillan group, for a reaction that I found particularly exciting, even while visiting graduate schools, where a hydrogen atom transfer reaction using, in this case, the quinacridinium radical cation is done to generate this alpha amino radical, where you get good selectivity for this position here adjacent to the amine because of the effect of this nitrogen-centered lone pair stabilizing the radical. Concurrently, a nickel 0 species that's present in the flask can undergo an oxidative addition into an aryl halide in a traditional step for cross couplings to afford this key nickel 2 aryl species. This can then undergo a step that was less common in the literature until recently, but has proven to be extremely useful, namely the capture of an alkyl radical by an organometallic species, which can afford these really interesting high valent alkyl aryl or alkyl alkyl, depending on the reaction species, which can then undergo a facile reductive elimination because of what a high valence what a high oxidation state you're in, to afford the key product in the nickel 1 species that can be brought back on cycle in a subsequent step. So what this enabled was a way to generally arylate these sorts of alpha amino CH bonds, which has been employed by our group as well as others for the synthesis of these arylated products at these specific electron-rich CH bonds. But what we were interested in was whether you might be able to do this instead of with these sort of specific, somewhat activated positions, just any generic CH bond. So something as inert as something like cyclohexane, which you would think about using as a solvent because of how unreactive it is. Could we use those as a precursor for radicals and then use them in conjunction with another metal to enable like CH to C aryl transformation? And while there are a number of methods that have been published in the literature in recent years that have enabled the functionalization of the arylation of CH bonds using, for example, the fact that they're acidic to deprotonate them and then do an arylation, or putting a directing group in the area to increase the effective concentration of your catalyst, there have been no general methods at the time we embarked on this project for the direct arylation of unactivated CsP3H bonds. So this was the scientific question that we asked ourselves at the beginning of my time in the Macmillan group. And based on the background that I showed previously with our ability to do this with specifically electron-rich CH bonds, one of the main questions we had to ask ourselves is whether we could actually do this key step where we're converting a carbon-hydrogen bond directly to an alkyl radical, and whether or not that step could actually be compatible with transition metal catalysis. If you think about what a high-energy intermediate you're generating here, are you going to be able to do that in a way that it's not going to interfere with a possibly sensitive transition metal cross-coupling cycle? So I won't go into great detail about the things we tried that didn't work, but suffice it to say that after thinking fairly broadly about this problem, we settled on this interesting structure here, the decatungstate anion, which might look quite complicated in the fluid dot structure, but I think is a little bit easier to digest if you look at the structure here, where it's just a bunch of tungsten oxygen octahedra stuck together. And this is well established in the literature in a really interesting range of inorganic and physical chemistry studies to be able to absorb light to enter a highly oxidizing excited state. And this is actually known to react at incredible rate constants. So in this case, it's about 10 to the 7th in the reaction with cyclohexane, which if you consider that just the simple rate of diffusion in solution is about on the order of 10 to the 9th, is an unbelievable rate constant for effectively ripping the hydrogen atom off of cyclohexane to generate a cyclohexyl radical in the reduced state of your catalyst. And while it might not seem as sort of impressive or interesting, another thing that was really key in our thinking about using this catalyst is that this 5- state is itself actually quite reducing. So it's minus 0.9 volts versus SCE, which might not mean that much other than that it means that if you have a reaction that also involves a challenging reductive step, this same catalyst that was oxidizing enough to remove a hydrogen atom from cyclohexane is also reducing enough to potentially reduce for example, a transition metal catalyst that might need to be reduced during the course of your catalytic cycle. And furthermore, it's actually known from great literature by the Yamase group in the 1980s that this itself, if it's just sitting around in solution, will actually disproportionate to generate an even more reducing species, which would enable you to have an even more reducing species in the flask that could do an even more challenging reduction. So while I don't expect everyone to have immediately digested everything on the slide, I think it really, to me, illustrates how cool working in the field of organic chemistry and more specifically photoredox catalysis is, because we were able to take this really broad range of experimental results that have been figured out by a number of very brilliant inorganic and physical chemistry groups over the last now almost 40 years, and see that this would be a catalyst structure that would absorb light in a convenient range, would be able to cleave strong CH bonds as we needed for our proposed transformation, and would be sufficiently reducing in its reduced state to possibly turn over the other part of the catalytic cycle. Now, one question that you might reasonably ask is if you have a catalyst that's capable of pulling the CH bonds off of something like cyclohexane, will it have any sort of selectivity, or will it just tear all your molecules apart? And while there's sort of, it would be reasonable to be concerned about whether or not this would be able to react with any sort of reasonable selectivity. What we actually know from work that has been done previously and from a lot of our experience doing other hydrogen atom transfer reactions in our group is that the electronics of CH bonds can give you a really good way to do selective hydrogen atom transfer functionalization. In particular, in this case, since the decatungstate abstraction occurs at a very electrophilic oxygen atom, it is very selective for electron-rich CH bonds. So in the case of something like tetrahydrofuran, the effects of these oxygen lone pairs adjacent to the CH bonds adjacent to the oxygen renders the CH bond particularly electron-rich. And in that case, a catalyst like decatungstate would be expected to have very good selectivity for functionalization at the alpha as opposed to the beta position of THF. The case is the opposite for something like cyclopentanone, where even though this position that's adjacent to the carbonyl is quite a bit weaker than the distal position because of the resonance delocalization you can imagine, the fact that it's adjacent to this empty pi star orbital of the carbonyl and has this extremely strong sigma electron withdrawing effect as well, renders the CH bond actually now significantly protic, not hydridic, meaning it is very electron poor. And your electrophilic catalyst then, for sort of polarity reasons, is probably going to struggle to abstract from that position. And you might actually be able to observe selectivity for the stronger but less electron poor CH bond. Finally, though perhaps less interesting, this catalyst is very large, as you can see just from looking at its Lewis structure. And because of that, you can also imagine getting selectivity based on steric effects. And at the time when we were beginning working on this project, there had been really great work done by the Fignoni and Ryu groups, where they had demonstrated in the context of a conjugate addition using a Michael acceptor shown here, that all of these electronic and steric effects were in play in the functionalization by decatungstate, where they did in fact observe the products you would expect based on these simple polarity models, suggesting to us that this structure could be used for selective functionalization in the way we hope to use it. So with that understanding of decatungstate and our background in metallophotoredox catalysis, we envisioned that the transformation that we were trying to accomplish would look something like this. Our CH nucleophile, something such as a cyclohexane, could undergo a hydrogen atom transfer reaction with the excited state of decatungstate to afford the cyclohexyl radical and singly reduced decatungstate. This cyclohexyl radical would be expected to react very quickly with a nickel species, in this case a nickel 0, to afford a nickel 1 alkyl. This could then undergo a facile oxidative addition into an aryl bromide shown here to afford the key nickel 3 alkyl aryl that appeared in the Princeton I showed earlier. This would then be expected to undergo a facile reductive elimination to afford the desired product and a nickel 1 halide species. And now at this point, if you look at the two catalytic cycles, what we need is this needs to give away an electron to regenerate the decatungstate 4-, and this needs to accept an electron to regenerate the nickel 0. And while based on cyclic voltammetry, this is not a sufficiently reducing species we would expect to reduce the nickel 1 down to nickel 0. We knew from our understanding of the decatungstate literature that this could actually undergo a disproportionation to afford the decatungstate 6-, and that the decatungstate 6- should be sufficiently reducing to reduce the nickel 1 down to nickel 0. And in fact, if you were to take a UV vis spectrum of the reaction, you can see that the decatungstate 6- is the resting state by UV vis, suggesting that it is the species responsible for the reduction of nickel. So with that, this project was a really great collaboration with a really fun group of people. One thing that's been great about working at Princeton is all of the close collaborations we have with chemists in industry. Danny Schultz and Dan DeRocco actually had experience working with decatungstate before we had started working with it, and were instrumental in particularly getting that area of research in our lab off the ground. This project was all started by Ian Perry, who did all of the initial optimization and a lot of the work on the CH scope of the reaction. And he was shortly thereafter joined by Tom Brewer, who is now a medicinal chemist at Genentech, who did all of the work that you'll be seeing on the aryl halide scope. So generally, when you're developing organic reactions, you have some brilliant idea for how it's going to work, and then you set it up and it doesn't work. But in this case, it actually was one of the few times I've ever seen where the way that it was drawn up and the way that Ian envisioned the reaction working essentially worked first time. I believe the first screen of this actually gave something on the order of 30% yield, and a brief optimization of just base and ligand gave about 70%. One thing that you'll notice looking at the one of the first things that we do when we're developing a photoredox reaction is we try and make sure that all of these components are necessary. If you look at it, they look like fairly complex reactions. We want to make sure that we're actually getting the sort of light-driven reactivity we expected. And as you'll see from this table, all of these components prove to be essential for the transformation, with blue LEDs working substantially less well than these 390 nanometer LEDs just because of the absorption spectrum of the catalyst. And as you'll see on this top line, we do use 5 equivalents of the CH component, which is not optimal. It would, of course, be great if you could use 1-to-1. But we did find that you could go down to 3 equivalents of the CH component with only a small decrease in yield, and could even use 1 equivalent as long as you were willing to accept a noticeably smaller yield. So with that, Tom looked at the aryl scope of this. So we wanted to see whether or not we had come up with sort of a general cross-coupling reaction that is tolerant of a broad range of aryl halides. If you see here, these were particularly interesting to us because of the kinds of protic XH, so in this case NH or OH bonds present in the molecules that are sometimes challenging for cross-couplings. We also were able to show that the reaction works broadly for electron-rich aryl halides, which are sometimes particularly challenging in that oxidative addition step. and aryl halides bearing an ortho group, which are also sometimes challenging due to the steric effect. Finally, we were able to apply this to a range of medicinally relevant heterocycles. These kinds of pyridines or pyrimidines or even these 5-membered heterocycles are quite common in the development of pharmaceuticals, and we wanted to make sure that this was a reaction that might be useful to medicinal chemists who would be trying to incorporate these sorts of structures into their drugs. But of course, if you look at the chemistry we've done to this point, we come up with a very interesting way to aerolate cyclohexane. But what we really wanted to do was see if we could use CH bonds more generally as the nucleophile component for a cross-coupling. And we were asking ourselves whether, based on our understanding of the selectivity of decatungstate, whether we might actually be able to do this selectively on these more complex structures shown here. Based on the arguments I presented before, due to the electrophilic nature of the oxygen-centered hole in the decatungstate, we should get selective abstraction of hydridic or electron-rich CH bonds. So sure enough, when we looked at the substrate like tetrahydropyran, where you have the 2 lone pairs on this oxygen rendering the adjacent CH bonds electron-rich, we were able to get a 55% yield of the arylated product at this alpha position without observing any arylation at the less electron-rich distal positions. Then if you look at the case like cyclopentanone, which we discussed before, where you have a weaker but electron-poor CH bond adjacent to the carbonyl and a less electron-poor but stronger bond beta to the carbonyl, we were in fact able to get complete selectivity for functionalization at the beta position, isolating this arylated product in 51% yield. Finally, we were also able to apply this to cases that look a little bit more complex, such as the 7-bromo-norbornane shown here, where the steric effect that I discussed previously prevents abstraction from these 2 positions on the same side of the ring as the bromine. The high bond association energy due to ring strain of these bridgehead CH bonds prevents abstraction at this site, and the electron-poor nature of the alpha-bromo CH bonds prevents abstraction at that site, resulting in complete selectivity for abstraction from these 2 equivalent positions on the opposite side as the bromine. And furthermore, the steric sensitivity of the radical capture step gave us what is complete exoselectivity and a 67% yield of this arylated product, suggesting to us that our simple model for the electronics governing the selectivity of this hydrogen atom transfer reaction was consistent with what we were observing with this transformation. So with this understanding of the selectivity in mind, we were able to apply this to an interesting range of particularly bicyclic CH bond bearing scaffolds to afford the corresponding arylated products in good to excellent yields. We were also, in what was some of the most interesting work to me, able to apply this to the direct functionalization of these interesting terpenoids shown here, which are actually, these both are used in, I believe, the fragrance industry, and it meant that my waste smelled quite good for a few weeks. We were also able to apply it to the functionalization of linear structures, such as hexane shown here, which, while perhaps not the most synthetically useful way to aerolate the 2 position of hexane, to me was of sort of interesting academic interest due to the large number of fairly similar CH bonds, and the fact that we were in fact able to observe good selectivity for this specific methylene position due to the combination of it being more electron rich than the primary position, but less sterically hindered than the 3 position. Finally, this did work, as I showed in the previous slide, for these sort of electron-rich alpha heteroatom CH bonds, which, while perhaps less interesting than these totally unactivated CH bonds, were something that this chemistry was particularly efficient for. So with this in mind, we wanted to illustrate how a medicinal chemist might think about using this chemistry. In this case, we took the bromide derivative of a top-selling drug, Celebrex, and what Tom did was he looked to see if we could append a range of different alkyl components to this position at Celebrex, starting with just the CH bond at these positions. And sure enough, Tom was able to, in a single step, using our decatungstate and nickel chemistry, directly attach either a cyclohexane, a cyclohexanone, or this interesting 7-bromo-norbornane unit at this position on Celebrex, resulting in a library of alkyl substituted Celebrex derivatives. Furthermore, we were also able to take the sesquiterpenoid scalariolide, and Ian was able to show, starting from a range of different aryl halides, or in this case, an acyl halide, synthesize a range of scalariolide analogs where we've now varied either the aryl or acyl group at this one specific position. We were also able to apply this to the shortest reported synthesis of this interesting non-opioid analgesic epibadidine, which, despite being incredibly toxic, was investigated as a scaffold for the development of non-opioid analgesics in, I think, the late 90s to early 2000s, with this compound here, terbanacycline, making it as far as phase 2. So we were able, just starting from the simple CH compound, to do a direct aerolation of that CH bond and access the Bach-protected derivative of epibadidine in 28% yield. This work was actually conducted at Merck because of the toxicity of this compound, and I don't think any of us were ever quite brave enough to remove the Bach group and synthesize the actual compound, but it was still the shortest formal synthesis of epibadidine. Finally, we were able to show that you could actually vary the aryl component, and this provided direct access to an interesting array of different heterocycle analogs of the epibadidine core structure. Finally, we were also able to modify the core, as shown here, to now make a bunch of different epibadidine analogs where instead of just varying the aryl substituent, we also varied this bicycle that forms the core of the molecule. So with that, I would like to thank Dave and all of the great coworkers I've had at the Macmillan Group for creating a great environment to work in for the last few years, and the really great team of people I worked on for the aerolation, Ian, Tom, Danny, and Dan, and the Ted Taylor family, Princeton University, and Merck for funding. And I'd be happy to take any of your questions.
Thank you, Patrick, so much for your talk. If everyone can please join me in giving Patrick a virtual round of applause. Just as a reminder, if you would like to ask questions, you can raise your virtual hand by clicking on the participants window in Zoom. If you're joining us from YouTube, you can send an email to the email address that's listed in the description box below the stream. Looks like we have one question from YouTube. Does the cross-coupling only work for CSP3H bonds? What about CSP2 hydrogens and CSP2 and CSP hydrogens?
That's a really great question. So this chemistry is generally restricted to CSP3H bonds, which comes down to both the electronics and strength of those different classes of CH bonds. So if you think about a CSP2H bond compared to a CSP3H bond, CSP2H bonds are actually substantially stronger and substantially more electron poor, because you can think of them as involving more orbitals with more S characters. They're more electronegative, closer to the nucleus. So functionalizing CSP2 and CSP bonds with hydrogen atom transfer chemistry is extremely challenging, because you're breaking a very strong CH bond and generating an even more reactive radical. So in this context, this chemistry is specifically useful for the functionalization of CSP3H bonds. And we thought this was also particularly interesting because there are lots of good methods for the aerolation of CSP2H, there are lots of good methods for the aerolation of CSP2H bonds employing different mechanisms, but the functionalization of CSP3 positions has remained generally more challenging in recent years.
That's interesting. Thank you so much. Are there any questions from the Zoom audience?
I have a quick question, Patrick. I was just curious if you see yourself going back to the medicinal chemistry following your graduation from Princeton. Did you like it a lot? Or?
Yeah, that's actually a really great question. I think the short answer would be that I did like it a lot, and that was my plan upon going to graduate school. Part of why I was interested in the group that I'm interested in is we have a really great history of placing people at really good industrial positions. But I've actually really enjoyed, particularly even the more esoteric parts of the academic work I've gotten to do here. So actually at the moment, my plan is to go and do a postdoc in maybe something more sort of inorganic or organometallic oriented than the work that I've done here, and then probably take it from there. But yeah, I've actually really, it's a really good question, and it's sort of a complex thing to answer because it's not that I didn't enjoy working as a medicinal chemist. I loved it, but I've also really enjoyed the work that I've done here and would like to maybe explore that a little bit. In part because a postdoc, I think, presents a fairly unique opportunity to learn different science in a way that I probably would not be presented at any other point.
Thank you so much. Looks like we have another question from YouTube. They ask, we're learning about radicals in first semester organic chemistry right now. Can you react at the benzylic or allyl position at all?
Yes, so actually, I don't think I have any examples in this. No, none of these are benzylic. But this chemistry actually works quite well for benzylic CH bonds. So as I imagine, based on the fact that you're asking this question, those tend to be weaker CH bonds because of the resonance stabilization of the resulting radical. And because of that, there are actually many more methods for the functionalization of benzylic CH bonds than sort of unstabilized CH bonds. So if you look back to the precedent that I showed at the beginning of the talk that really inspired a lot of this chemistry. So this reaction here, This quinoline system that was used previously actually does work for the functionalization of benzylic CH bonds as well. So both the decatungstate chemistry as well as other systems can do CH or benzylic CH bond functionalization. Allylic bond functionalization is actually sometimes a little bit more challenging because there's also a lot of sort of ability of radicals to react directly with alkenes. So in that case, this decatungstate chemistry has generally not worked particularly well, and we think part of that might have to do with polymerization because of the radicals adding into the alkenes. But actually, the chlorine radical and bromine radical based conditions do tend to work fairly well for allylic functionalization as well.
Awesome, thank you so much. Are there any questions from the Zoom audience? Okay, well, you guys think of questions. You can also type them in the chat if you don't feel comfortable unmuting yourself. So I have another question from YouTube. Do you have to come up with research projects in graduate school? How did you choose your research?
That's a really good question. That's something that is in some ways kind of challenging to answer because it can vary a lot even for one person from project to project. So I think it also will vary a lot. So it will vary within a person at a specific research group. It will vary a lot from research group to research group. So different advisors will have different approaches to the right way to address this problem. I would say that the short answer is that you don't have to feel like you need to arrive at graduate school with a bunch of ideas that you want to work on. There are some people who do that, but obviously since they're ideas that you came up with as an undergraduate, you're going to learn things in your graduate studies that might help you to come up with ideas that are maybe slightly more likely to work. So it's, I guess, like a back and forth process where the way it worked for me is I arrived and there was a general idea that my advisor, Dave, was particularly interested in. So he basically presented, here's a transformation that we would like to do. And then it gave me a list of people who would be interested to talk to about it and some interesting resources and said he would be happy to chat about it. But it's sort of the general concept was something that my advisor was very good at steering me to. But the sort of day-to-day details are something that you spend, particularly for your earlier projects, a lot of the time figuring out. And the way that I chose my research, I guess it would depend if that is sort of more general, like I chose this research group, or if it's like the research I did within this group. But if it's sort of the more general question about why I chose the kinds of research that I wanted to do in my PhD, which is photoredox catalysis, it would be because I really like the way it shows the interrelation of seemingly esoteric, very academic concepts, like the absorption of light and the relative stability of radicals, how it uses those sorts of concepts to develop reactions that can be transformative for being able to make drug-like molecules. It's just like, I think it's a really cool way to put together very academic understanding of chemistry and use it to develop something that is very useful for people who want to cure diseases.
That's so interesting. Thank you so much. Are there any questions from the Zoom audience? Feel free to raise your virtual hand or type the question in chat. If not, I do have another question from YouTube. Why doesn't the nickel react with the bromine on the norbornane ring?
That is a really good question. So the short answer is that is a very specific alkyl bromide. So if you think about the ring strain that's present in that system and the way that the 2-2-1 bicycle is sort of blocking the sigma star of it, it makes it a very challenging thing to access to sort of directly functionalize in a 2-electron pathway. So that sort of bromide is actually quite bad at doing the SN2 reaction. And if you think about your nickel oxidative addition instead occurring through what would be thought of as like a halogen abstraction reaction, where instead of it attacking the halogen from the backside, you're actually pulling the halogen off to generate the resulting alkyl radical. That one is actually quite challenging as well because of the fact that this makes, due to the ring strain, a very unstabilized radical. I think part of it also just has to do with the fact that generally the oxidative addition into aryl halides is quite a bit easier than oxidation into alkyl halides. So the fact that we have a stoichiometric amount of aryl halide present just means that we do probably outcompete it. Though it is interesting, we don't see any bromine functionalization or bromide functionalization in that, or alkyl bromide functionalization in that case.
Awesome, thank you so much. Are there any questions from the Zoom audience? You guys don't have to be shy. You can also type it in the chat and I'll read it for you. If not, I would like to thank Patrick for his amazing talk. Will everyone please join me in giving him another round of applause? And then with that, I will introduce Tiffany, our next speaker.
Thank you so much.
Of course. Tiffany is currently a fifth-year graduate student in the Macmillan Lab at Princeton University, where she is also doing research in photoredox catalysis to create alpha-aryl carbonyl motifs. Before going to Princeton, Tiffany got her first bachelor's degree from Harvard. She then went on to UCLA, where she conducted research in the Halk Lab on computational chemistry, and then went on to UC Berkeley, where she earned her second bachelor's degree in chemistry. At UC Berkeley, she worked in the TOST lab on boron catalyzed reductive elimination reactions. This research introduced her to catalytic chemistry and led her to Princeton. With that, please join me in welcoming Tiffany.
Thanks so much, Amanda, for that comprehensive introduction. For my background, I am, like Amanda mentioned, my name is Tiffany. I'm a fifth-year graduate student in Dave's lab, and I'll be talking. About, as you just can probably tell from her introduction, I have a very my winding road and like the lessons I learned along the way to get to where I am today. So I basically wanted to start out by saying that normally you would expect that, you know, when you're thinking about your future, generally you envision that you're going to take us like a straight path directly to like your permanent job. So you're going to graduate from high school, go to college, then go to grad school, and then get a post-grad job. But normally this just seems like it's a straight shot, and this is something that can seem like a really straightforward thing. But that's what I thought, and my reality was completely different. I actually had like a kind of like a pinball machine like trajectory where I did not at all go it did not go at all the way I thought I was going to go. But it actually turned out that these detours were really critical to helping shape the trajectory of my chemistry career, and every single step helped me figure out what I wanted to do in the next step. And I hope my takeaway from my talk will be that that it's okay to explore, and it's okay to kind of try to, you know, what might look like mistakes actually will turn out to be really important, like stepping stones to your next step. So just keeping that in mind as I go along. And even when I did decide to do chemistry, I actually bounced around within the field of chemistry within different subfields as well. So once you start doing chemistry, you don't have to necessarily stick to one field in your academic studies as well. So I guess unlike Patrick, I actually am able to use this map because I've actually gone a few times around the country. So I guess I'll start my story. We're in Southern California, which is where I was born. So I was born in this beautiful town called Moore Park, and it was like a 20-minute drive to the beach, you know, an hour drive to LA. So it was like a fantastic place to grow up. And I was really lucky to have a lot of opportunities. I loved like music as a kid. I did like a lot of piano, violin, and I seriously considered music school. And I loved also literature and writing as well. So there was I did not expect to be a scientist by any means, although I did have some small interests in like things like archaeology and chemistry. And I had a fantastic AP chemistry teacher named Tim Hogue. So he inspired me I had that in the back of my mind all throughout college. So that was just something that was really fundamentally inspiring to have him as a teacher. So as a result of these different like interests, I really, so instead of going to music school, I realized that maybe I wanted to get like a more holistic kind of education for my college degree. So instead of applying music school, I actually applied to just regular colleges. And I wound up flying across the country to Massachusetts, where I went to a small school outside of Boston called Harvard. And then I couldn't decide whether I wanted to do humanities or STEM major. So basically this entire journey, I had this entire journey in my first 2 years of college where I was very unsure what I wanted to do. So eventually I landed in a field called biological anthropology, which is essentially ask the question, how did evolution make humans the way they are? So we study like a lot of things like why people run, and how fire helped people evolve into people, basically, in the science of cooking and things like that. And then I also had a minor in literature, so that humanities background is always in the back of my head. So, and also this major it seemed like a perfect fit for me because it actually bridged humanities and science. So I actually did coursework in social anthropology, human evolution biology, and archaeology. And in fact, I actually I did archaeology field school in my second year in college, where I went to this really interesting Mayan ruin called Copan Ruinas in Western Honduras. This is our dig site, and this is me brushing off some dirt or like a centipede or something off of a rock here. It was really, really fun, but also really, really hard. There's a lot of digging and a lot of confusing pot rocks for pot shards, and it's it was a journey for me. But this was a fantastic experience, and I really appreciated being able to just have this great summer learning how archaeology works. But I realized after this experience, this is not something I wanted to do in the future. So I joined something called the Nutritional Ecology Lab, which is which is more in the biology field of advanced biological anthropology, where we basically studied these like diet samples from this hunter-gatherer tribe in Tanzania called the Hadza. And they basically are one of the few remaining hunter-gatherer tribes in the world currently, and so they have tubers and a lot of berries in their diets. And then my job in this lab was to basically do a lot of analytical chemistry and sit in the lab and kind of figure out things like nitrogen content of these samples. And in the course of this, I realized I really liked being in lab. I really liked thinking about molecules. I really liked doing lab work and like the precision in the precision involved in doing lab work. So this was just kind of a really good entry into like the field of kind of like the idea of like doing lab work in chemistry in the future. And I always had that in the back of my head going forward as well. So after this, I completed my my degree in anthropology, and then I graduated. And then so of course, what I wanted to do was I fly back across the country to California, where I flew back to Southern California, where I just got a job at a small startup, and I just worked as a project manager. In that time though, I based on my chemistry my interest in chemistry was I just read a lot about chemistry. I like to read books about food science and things like that. So I wound up deciding to in my spare time take some organic chemistry classes through UCLA Extension, which is within like commuting distance from where I was living at the time. So this was a really momentous decision, because like this led me down a complete route that was completely different than what I thought I'd be doing. I thought I'd be a desk job for the rest of my life, and that did not turn out to be the case. So when I went to UCLA, I took some classes, and then I was really fortunate. I was really fortunate because I reached out to some professors who was interested in doing research. So I reached out to Professor Kendall Hauk, and he said yes to me joining the group to do some research. And I joined this. The Hauk group is a computational chemistry group at UCLA. Basically, they use quantum mechanical methods such as density functional theory and things like molecular dynamics to study the mechanisms of like organic, organometallic, and biological reactions. So this was kind of different. This was kind of different than what I did, what I'll be doing, what I do now. But so this was a completely. I started out in a completely different field, essentially. In this lab, I was really lucky to work on 2 different projects. In a subfield that in a subfield of organic chemistry that the Hauk group is really known for, which is like studying the mechanisms of cycloadditions. So in both of these projects, I studied basically Diels-Alder and higher order cycloadditions. So I'll talk about that in the coming slides. So when I first joined the group, I studied these. I basically studied these things called transannual Diels-Alder reactions. So just to give a little bit of background, you guys have learned about intermolecular Diels-Alder +2 reactions. So if you add like a tether to it, if you add something like just some carbon chain here, this then becomes an intramolecular Diels-Alder reaction where you have a second ring appended to this initial ring. If you have 2 tethers, this becomes a transannular Diels-Alder reaction where basically you generate 3. It becomes a tricyclic product. And so these generally react very quickly at room temperature. And these tricyclic products generate a lot of complexity in one step with some predictable stereochemistry at these ring junctions. So these are really interesting reactions to study and really hard to study experimentally because of how facile they are. So these are perfect to study through computational chemistry. This is something that this is a complete coincidence, but the reaction that I studied computationally in this project was actually Professor Ife's PhD thesis. So if you want to know more about it, you can feel free to reach out to Professor Ife and talk to him about this reaction. But this basically basically what they did was they did a cross-coupling reaction to try to cyclize into this macrocycle. but they were never able to isolate this macrocycle. And instead what happened was that they immediately would just immediately undergo the 4+2 Diels-Alder, transannual Diels-Alder to generate this product. So as a result, this indicates that this cyclization is incredibly fast at room temperature. So I was assigned to I worked with Dr. Cindy Ho, who's my graduate mentor in the lab. She was a fantastic mentor, and basically she taught me all the ropes to how to do research in the Hauk Lab and just research in general. So I'm really indebted to her for all of her mentorship throughout my time at the Hauk Lab. And basically what we did was we got some experimental data from the Merlitt group on these transannual Diels-Alder reactions. And she taught me how to use this thing called the distortion interaction model, which is something that the Hauk group is really well known for in computational chemistry, which basically, if you think about a reaction coordinate, it breaks this model kind of tries to understand the factors that contribute to reactivity by breaking down the activation energy into something called a strain activation energy and an interaction activation energy. And by looking at these different types of energies, you can try to understand the different factors that lead to this transition state. So this transition state will occur when the activation strain that you see here, which is here delta E diagonal strain, is overcome by the stabilizing interaction, which is usually an electronic interaction which is indicated by this delta E double dagger here. So the changes in these energies along this reaction coordinate, as it is written here, give insight into the factors controlling reactivity. And the factors we looked at were the tether length, so like the length of these tethers on the sides of on these rings, and the types of heteroatoms that are in the rings, and the types of dienophiles, so either alkenes or alkynes. So basically, we used this model to generate a lot of data on the activation energies and the strain and interaction energies of this reaction. And we compared them to the intermolecular and intramolecular Diels-Alder data as well. And just to summarize briefly, so I don't get too much in the weeds here, the main conclusions we drew were that the reactive transannual Diels-Alder substrates, the most reactive ones, have lower distortion energies because of the tethers pre-strain. The tethers pre-strain the ground state pretty much. So these are already initially strained, so it makes the barrier significantly smaller. And we also looked at different ring sizes, and we found that the 12-member rings have the fastest transannular Diels-Alder due to favorable enthalpy. So just some facts for you to keep in your back pocket if you ever need that. And so just for these, as a summary though, this project was really, really important to me because I learned that, I learned from Cindy Ho, who is my mentor, and I learned that research is very much kind of a learn as you go. Like, you're basically at the frontiers of science, essentially, in a research project. So as you're going, it's going to be really scary because there's going to be a lot you don't know. So you have to be constantly comfortable beyond like the edge of your knowledge and kind of being beyond your comfort zone. So that was kind of like the key takeaway for me from this research project, was that research is kind of like this kind of scary ride where you don't really know what's going on, but as you go along, you have to kind of it's exciting, and it's exciting, but it's also very learn as you go. So for those of you who haven't done research yet, that's kind of the experience that I had, at least in my first project. So this was foundational because then I was really fortunate to be given an independent project by Professor Hauk to spearhead. So this project was a really fun project. So just to build on what we just talked about, we know about the Diels-Alder. So how about higher order cycloadditions? And by higher order cycloadditions, what I mean is, what if you have like a 4-pi and a 6-pi system, so such as this fulviane and a tropone? So these 6+4 cycloadditions, as you can tell, are thermal. You learned the Woodward-Hoffman rules. They're thermally allowed, and so when you heat this will lead to the formation of these 10-membered rings, and based on the relative, based on they can be either exo or endo in the conformation of the product. So this was predicted by Woodward-Hoffman in 1965. So to give you some background. So as you can see from this orbital diagram, this MO diagram, this is predicted to be orbital symmetry allowed under thermal conditions. And this is actually experimentally validated in the next year by these two researchers, Cookson and Ito. So, Professor Hauck, just to have some historical context here, he worked with R.B. Woodward at Harvard for his PhD. And when he did his PhD work, he actually did it on this exact reaction. He worked on this, this is 2 years after they proposed that this was a feasible reaction, and he discovered in the course of 1967 that when you run this reaction and heat it in THF, you generate this 2-to-1 adduct of tropone and dimethylfolvine. So you have 2 tropones and 1 dimethylfolvine here. So this was back in the day when there wasn't a lot of really complex structure, complex technology. So it took them a while to figure out the exact structure of this molecule. But in 1970, through some help by some NMR experts, they were able to elucidate that this was the actual structure of the molecule. But this is actually a really complex reaction, because if you actually run this at room temperature over 3 weeks, you get a completely different distribution. You get some of this product instead of quantitative amount of this product, and you get a mixture of other 6+4 cycle addition products. So there are a lot of considerations here. There's a regio selectivity, stereoselectivity, as well as this thing called periselectivity. And periselectivity is this thing. In periselectivity, I'll talk about in a slide. But the mechanism that was proposed for this reaction was you have this these 2 adducts. They undergo a 6+4 reaction to generate this kind of intermediate, and this can undergo a 1,5 hydride shift, a sigmatropic shift of this hydrogen to generate basically this product. And then this can undergo another 6+4 cycle addition with a tropone to generate this the final 2-to-1 adduct. So this is just a hypothesis. So based on the complexity of this hypothesis, we thought this would be a really good reaction to study through DFT. So basically, this reaction has really complex. So when I mention periselectivity, what I mean is that you can actually generate a number of different types of adducts using in this reaction when you heat it. So you can have the 6 pi system be the fulvine, and this is borne out in these 2 types, these two diagrams, or you can have the 6 pi system be the tropone, which is borne out in this diagram. But you can also have 4+2 reactions that happen, and the 4+2 reactions can then also have tropone as either the 4 pi system or the 2 pi system. So you can imagine that this then presents a lot of complexity in terms of understanding this reaction, and these are all structures we had to calculate and to get like the energy. So these are all the relative energies of these structures. So this was a lot of brute force work, and it was a lot of fun, but it was also like a lot of work to try to understand what's going on here, because there's just so much stuff to look at. But I think the most exciting part of this reaction was that at some point, so with the help of a lot of mentors in the group, we figured out that I kept getting these errors in my in the calculations, and it turned out that the transition state that I was trying to calculate was actually something called the ambimodal transition state. And what that means is that this transition state, so this is TS1 would be like this point right here. It means that this can directly lead to 2 different adducts depending on just the dynamics of the reaction. So this transition state can actually either go to this product or this transition state can go to 2 different products, and then that it becomes really complex. So this is like a really flat surface to try to calculate basically. So this is a really exciting discovery, because this is something that this is just something that was it's been known before, but basically we had to calculate. We found that a bunch of different types of ambimodal transition states in this reaction, and by comparing all the different energies of these transition states, we were able to eventually elucidate a proposed mechanism for this reaction. So just as a really brief overview, the first proposed cycle addition goes to this exo product, and you don't need it. I'm not expecting to know the details here, but basically it goes to this exo product, and it undergoes a Koper rearrangement to get to this product, and then it undergoes a 1,5 sigmatropic rearrangement to go to this product, and then it undergoes another 6+4 cycle addition to generate the 2-to-1 adduct. So basically, in summary, this is just a really complex reaction, and there's a lot going on. But it was incredibly challenging to work on this project, but also incredibly informative, and it kind of built in me this kind of love of this excitement for discovery and kind of studying the mechanisms of really complex reactions. Additionally, this project was really interesting because it taught me how to start and develop a project from like kind of start to end. So it was a really kind of a really fundamental, this is a really really good experience to as an undergrad to try to spearhead your own project. Sorry, this is frozen. So yeah, apparently my computer thinks this is so complex that it just decided to freeze. Right. Yeah, this is okay. So with that, so yeah, so with sorry, computer is being really slow right now. Okay, so with that in mind, I next, so this was all a computational work, and I really was interested in also, I was very curious about the synthetic chemistry. So I thought that it would be really important for me to try to understand what my interests were, and completely to understand what my interests were before I applied to graduate school. So I was really lucky in the sense that I was able to, I found a 2-year bachelor's degree program at Berkeley. So this is my shortest move, but I moved from UCLA to Berkeley right after I did my work in the Hout group, where I wanted to learn how to do synthesis in this 2-year program. And I wanted to also get a better foundation for graduate school in terms of like coursework. So I wound up joining the TOST lab. This is Professor Dean Tost, which develops this lab, the lab's research develops catalysts and catalytic reactions. A lot of them are using it through are for gold catalysis. And my mentor in this group was Mark Levin, who was really, really talented, really talented graduate student, and now he's currently an assistant professor at University of Chicago. So anybody who's interested in applying to University of Chicago should definitely apply for Mark, because he's a fantastic mentor. So I worked initially on an independent summer project on the formation of HO-isomers using gold catalysis, but that never got off the ground. So this was another lesson in that projects often do not work, and that's just going to be the reality of graduate school in research. But so I did learn a lot in the process, though, because I spent a lot of time in this thing called the glove box. I'm making a lot of gold complexes. So this is basically a big nitrogen filled or argon filled box that allows you to do really air sensitive chemistry. And in these gloves, you just stick your hands in them, and then you manipulate your chemistry in the glove box so that you can exclude any air. And it was really, really fun and kind of tedious, but this is a really good, this is just a really classic organometallic chemistry kind of environment for those of you interested in doing inorganic or organometallic chemistry. Some people love it, some people hate it. But, so in the course of my time in the TOST group, we worked on a couple of reactions, and then Mark found this really serendipitous result where he took this gold complex, and when he added this borane, he actually found that this induced reductive elimination of methyl CF3 and the formation of gold 1CF3 complex. So what this told us was that this was actually a completely novel thing. So I worked with Mark to study this reaction mechanistically to understand the distinctly distinct fluoride abstraction migratory insertion mechanism. And in the course of this reaction, in the course of this project, I learned a lot of things. I learned how to do stoichiometric studies, stoichiometric studies for mechanistic studies, something called an Irene analysis, and also I used some DFT in this reaction as well. So this was a really diverse project. And just to walk you through really briefly, I'm not expecting this to be really easy to understand just at a first glance, but you have this borane can undergo abstraction from this this gold 3 complex to generate a basically a difluorocarbene, and then this difluorocarbene then can undergo migratory insertion into this adjacent alkyl group to generate this this fluorinated alkyl fragment. On reductive elimination, you regenerate this borane, this catalytic and borane. So either you have to figure out this migratory insertion was operative, and through a lot of stoichiometric reactions, this is basically the mechanism we came up with. So the critical thing of this reaction was that Mark realized that this fluoride rebound mechanism could allow us to do radio labeling with F18. And this radio labeling has important biomedical applications for the positron emission tomography tracers for the real-time imaging of tissues. So those are just PET scans that you have probably heard of. So this was the insight that Mark had that made this paper really impactful. So this whole project was really fun to work on, and it made me realize that I'm really excited to study catalysis and reaction development, and just the synthesis of gold complexes and just kind of understanding how they work and all the different mechanistic steps involved in this reaction was really opened my eyes to the fact that I really wanted to work on synthesis and methodology in graduate school. So that brings me to basically Princeton. But before I talk about that, I'm just going to discuss briefly the things that I learned before I started applying for graduate school. The first thing that I talked about with my mentors was, what do you want to do? Do you want to go to graduate school? You want to consider like, what do you want your lifestyle to be like when in your professional career? And I found that I really liked the freedom of academic research life. I really liked the freedom to pursue my own research interests, and I liked collaborating with and like learning from all these like scientific colleagues. And just my work, my research like experiences like led me to really kind of hone down my professional goals to, I realized I wanted to do an academic career. Just talking to my mentors and just talking to like Dean and Ken and a lot of chemists, I realized that this was something that I wanted to do. So with this in mind, I decided that yes, I need to pursue, in order to get to this professional goal, you need to pursue PhD. So grad school is the logical next step. So I applied to a bunch of places, and eventually I, of course, decided to choose a graduate program that was across the country once again, so I could move across the country. And I decided on Princeton. And so the reasons why I chose Princeton, and this is hopefully this will help for people who are thinking of graduate school applications this year. So basically I went to the visit weekend, and I looked at the program, and I knew that I wanted to do catalysis, and I looked at the faculty members who were at Princeton. I was specifically interested in photoredox catalysis after having when you as an undergrad you want to read a lot of breadth in terms of your reading. So I was really interested in radical chemistry and photoredox catalysis. And this department, and the particular Princeton's department, had a number of faculty members are working in this field. So there's Dave, Rob Knowles, Todd Heister, and Abby Doyle. And I knew that I wanted to go to a program that when you choose a graduate school program, you should always choose a program that has multiple faculty members you're interested in working for. So I knew that I'd be happy working for any of these professors. So this was this was an important consideration for me. And the final consideration was that when I went there, not only was the department really well funded and had a lot of resources, but the people there were also fantastic. So I got along really well with people in the Macmillan group, and also just generally in the department. And this is a picture of our group at our first Frickmas party, which is a Christmas party we have every year in the department, and just having a great time here. So I met the group, and they were really just a fun group of people, but also really invested in each other's success. And this was a really important part of the group culture, and that was what drew me into Dave's group in particular. I was also really excited about the research, of course, and then I was also really comfortable working with Dave as an established PI. So these were all factors as to why I chose Dave's group. And considerations for your grad. So basically, considerations for your grad school lab should basically really look at the research and the people you'll be working with. Oh, just to wrap up this section. So as you've seen, I've gone through a lot of different types of chemistry and types of my trajectory hasn't been very straightforward, but these might look like irrelevant detours, but each detour built in the last. So at Harvard, I realized I like lab work. Just to summarize, at UCLA, I realized I like mechanisms, but I also I wanted to try synthesis. At Berkeley, I realized I liked synthesis and wanted to try methodology. And then at Princeton, now I'm doing methodology, and I realized that I want to apply those methodologies to materials chemistry, which is where I'll be going next. And just to reemphasize that it's totally okay to not know exactly what you want to do when you start out in chemistry. You can definitely, you can and should just try different things and bounce around and try to figure out what it is you like. And it'll give you a good skill set to go forward. For example, now I'm pretty familiar with computational chemistry, and I try to use that in my research here at Princeton. So just trying different things will let you build a good foundation for your future research. And also my last thoughts on this is that my humanities and STEM are not mutually exclusive. Like I had a lot of, my humanities background taught me a lot. It taught me that critical thinking, it taught me how to critically read texts and how to read dense and long texts. It taught me how to create a narrative for my science, which is really important when you're doing research. Being able to craft like talks and craft papers requires you to kind of tell a story about your science, and that's really important to communicate your science. And then another thing that humanities taught me was that you can look at your science from multiple perspectives, which is really important when you're thinking about how to take your project to the next level. And then finally, some main lessons I learned on the way were just to keep an open mind and make some curiosity-driven decisions, be persistent in your goals, and then also the most important thing was finding advocates and mentors in your career, because it really takes a bunch of people help and support to get you through graduate school and beyond. And it's just incredibly rewarding to work with these really talented chemists. Okay, so that brings me to Princeton, which is I'll be talking now about my research in the Macmillan Lab. And as you've heard through the course of the seminar series, in the Macmillan group, we utilize photoredox catalysis to do chemistry, specifically to enable single electron transfer or energy transfer between photoexcitable catalysts and organic molecules via visible light. So this is in our reactions, we set up our reaction vials in front of these lights, these blue Kessel lamps, to do photoredox catalysis. In these vials, we have photoredox catalysts, which are able to do oxidation reduction or energy transfer to and from organic molecules. So we use these catalysts because they absorb in this blue visible light region from ordinary light bulbs. And so this allows us to target the delivery of energy via selective excitation of this photocatalyst to organic molecules instead of directly exciting the molecules with high wavelength, short wavelength light. And as a result of photoredox catalysis, we're able to generate these radical intermediates from really abundant and stable functional groups to engage in these valuable transformations and to activate small molecules. So in my research, one of my projects in this group has involved the formation of alpha aryl carbonyl complexes. And this motif is a really key functionality in a lot of biologically active compounds. So you may know of naproxen or ibuprofen, and these are also known as Aleve or Advil. So this is really fun. This is really important motif in non-steroidal anti-inflammatory drugs. And they're also found naturally in things like alkaloids in nature. So this motif is incredibly important in biologically active compounds. And there's also a wide range of different types of carbonyl compounds that are found in these active compounds, including free carboxylic acids, amides, and ketones. So the classical way to make these alpha aryl carbonyl motifs is through enolate arylation, in which you take an aryl electrophile and a metal enolate, such as an alkali or zinc enolate, and through metal catalysis, you can generate this alpha arylated compound. This is this canonical work was first done by Mura, Natsume, Buckle, and Hartwig, and it's an incredibly powerful reaction that you will definitely encounter in the course of graduate studies if you pursue them. So just to walk you briefly through the mechanism, this will start out with starts out with a palladium zero complex catalyst that undergoes oxidative addition into an aryl bromide. So and then you generate an enolate through the deprotonation of some sort of a carbonyl compound. And this enolate will then undergo transmetallation onto the palladium to generate this palladium enolate, aryl enolate species. In order to declination, you form the desired alpha arylated product. So the key components of this reaction are a metal, a ligand, and a base. So some common design issues are that you want to choose your ligand and catalyst system carefully to prevent the over-arylation of your product. This will also affect the beta hydride elimination step, and also it'll mediate the different rates of every elementary step. And also the choice of the base is important to allow for the formation of the enolate. So you can imagine that the arylation of nucleophilic enolate equivalents can be done on all these different types of alkyl nucleophiles as long as you can generate these enolates through using your base. But these different classes of alkyl nucleophiles have significantly different pKa's, ranging from 27 to 35 for esters. So as a result of this, the reaction conditions variables, specifically the base catalyst and ligand system, will significantly rely on the nature of the coupling partners, specifically the alkyl nucleophile. And additionally, you can imagine that protic functionalities such as carboxylic acids and amides simply can't be tolerated in these conditions, because you will just directly deprotonate these functionalities, and these will interfere with the desired reaction. And then finally, pronucleophiles such as toluene simply can't be deprotonated using just commonly used bases in organic chemistry. So you're not able to access these types of functionalities under these conditions. So you can imagine that these different carbonyl compounds will then require divergent conditions for enolate aerolation. But if you can actually, so the question I want to ask was, can you actually develop a potential alternative through an open shell intermediate as an enolate surrogate? So this open shell platform will allow us to basically have agnostic reactivity with respect to the carbonyl substitution. So you can use the same set of conditions for all these different types of classes instead of tailoring one set of conditions for every single class. Additionally, radical chemistry, as you're probably learning in classes right now, are able to be used in pretty mild reaction conditions. And finally, this type of reactivity would be orthogonal to like the classical methods I just talked about. So these are the motivations for why we wanted to look at this reaction. So the key question I wanted to ask was, can you actually generate these what are called alpha acyl radicals with similar reactivity across these diverse carbonyl classes? And once you generate these alpha acyl radicals, can we develop a platform to once this loads? Can we develop a platform that will allow us to access a wide range of alpha arylated products using a common set of reaction conditions? So this is gonna be a little bit slow right now. So basically, yeah. So we just to okay. Sorry about this. I hope it doesn't crash. There we go. Okay, so great. So the question I want to ask was, can we develop a novel and general open shell approach for alpha aerolation to generate a diversity of alpha arylated products? So I guess while we're waiting for this, this project was when I started in my first year. Oh wait, and I started this project alongside my mentor in my first year, who's Dr. Chip Lay, shown here, and Dr. Patty Chen, who was also a former graduate student in the lab. And they developed this really interesting cross-coupling reaction that utilizes what's called a cross-electrophile coupling manifold, where you have an aryl bromide and an alkyl bromide, and through photoreactive catalysis and nickel catalysis, you can generate this sp3, sp2 bond. And this works by the formation of basically an alkyl radical. This key alkyl radical intermediate is formed by the abstraction of a halide from this alkyl bromide using the silicon-centered radical. And the silicon-centered radical is relatively nucleophilic, and as a result, it's polarity matched to this. It's polarity matched in this abstraction step. So this is why this goes, and I'll talk about this a little bit more in a couple slides. So the question I want to ask was, can we bypass the need for enolates into electron chemistry using a class of alkyl electrophiles that are shown here? These are activated alkyl chlorides. And the reason why I chose this class of electrophile was because these activated alkyl chloride carbon-chlorine bond dissociation energies fall within a narrow range, within the mid-70 range. As a result, you can imagine that the abstraction step, where you can do halogen atom abstraction using a solid radical, should be kinetically similar across these different classes of alkyl electrophiles. So this might allow us to use a common halogen atom abstraction to form open shell intermediates. And because once you generate this radical under kinetically similar rates, then this radical should then be able to be intercepted by a nickel catalyst to generate a nickel 2 aryl species and allow us to access the general method for alpha aerolation. So just to reiterate the mechanistic design for this reaction, the alpha aerolation undergoes, this is an alpha aerolation via a single electron manifold. And as I just described earlier, this can be generated through the abstraction of a halide using the solid radical. the silicon-centered radical abstraction is thermodynamically driven based on the differences in the silicon-chlorine bond dissociation energies and the carbon-chlorine bond dissociation energies. So this bond dissociation energy is about 113. As we just saw, this is about 70 something. So this is very, very downhill. And this allows us to generate this radical irreversibly. And this is kind of the critical part of this mechanism. So as I just described, and just in mechanism here, we can talk about as the mechanism we propose is that you can excite your photocatalyst to generate this oxidizing triply excited state of the iridium. Then this can undergo a single electron transfer from a bromine anion to a bromine radical, bromine anion to a bromine radical. And then this can undergo HAT or hydrogen atom transfer from a supersilane to generate the silicon-centered radical. The silicon-centered radical can then undergo halogen atom abstraction to generate this alpha acyl radical, as well as the silicon chloride, this acyl chloride that we just talked about. So at the same time, you can have basically a nickel complex, a nickel zero complex that can undergo oxidative addition to an aryl bromide. And this can generate this nickel 2 aryl species, which can then trap this alkyl radical to generate this nickel 3 aryl alkyl species. And then on reductive elimination, you will then form a nickel 1 complex, as well as the desired alpha arylated product, which will show up here. Okay, and then this nickel 1 can then be reduced to nickel 0 by the photocatalyst. And in doing so, this turns over both catalytic cycles. So, and so the nickel complex we use is this will show up in a second, but it's this nickel ditriotyl BP complex. We use this classic iridium DFCF3 photocatalyst, as well as a silicon cell radical precursor. So in the course of developing a reaction, you basically start out just by using your best guess. So the best guess that we had was using the conditions that Chip and Patty developed in their reaction. So we were really lucky in the sense that by using those conditions and using this alkyl chloride, we were able to get an initial hit, about 33% yield. So this was about like October of my first year, and this was a great result because initially I was seeing 0 initially using the alkyl bromide. But once we got this initial hit, we next did what's called optimization, which is basically taking all the variables and trying to change them to try to increase the yield of the reaction. And so when I changed the base to, this is 2,6-lutein, which is a soluble organic base, we saw an increase in the yield because of the increased solubility of this base. When we increased the concentration to 0.5 molar, we saw an increase in yield as well, because this allowed us to, we think this allowed us to minimize solvent coupling. And then when we increased the nickel catalyst loading, we also saw an increase in yield because we thought this trapped the alkyl radical at a faster rate. So with these conditions in hand, we got, with these optimized conditions, we got a more optimized yield of 65%. But the major observed byproduct we saw in this reaction was protohalogenation of this activated alkyl chloride. So what that means is that we thought that this was being basically reduced by this process in which the silicon-centered radical would undergo the halogen atom abstraction to generate this alkyl radical. And this alkyl radical can then also undergo a reverse hydrogen atom transfer from supersilane to generate this proto-dehalogenated alkyl byproduct, as well as this chain-carrying silicon-centered radical. This silicon-centered radical can then go on to undergo further dehalogenation of these alkyl chlorides. So with this kind of hypothesis in hand, we kind of wondered, can you actually use a different silyl radical precursor to minimize this alkyl halide dehalogenation step here? And indeed, when we actually changed the silicon-centered radical precursor to tris-triethylsilosilane, which basically is known to have a silicon-centered radical that is more sterically encumbered through what are called EPR studies. We saw a significant boost in the yield. So because we think that this radical is more hindered, this is less likely to undergo it undergoes the halogen atom abstraction slower, and that allows us to match kind of the rates of the different catalytic cycles. So so we're really happy to find that by using this tris-triethylsilosilane radical precursor, we were able to get our highest yield yet. So with these optimized conditions in hand, we next turn to looking at what's called the substrate scope, which is something that in the course of methodology development, it's really important to be able to allow other chemists to understand what types of functionalities are tolerated in this reaction. So in our substrate scope, we found that different types of hetero-aryl halides, so changing the aryl bromide partner, we were able to tolerate a lot of different types of coupling partners. So we were able to tolerate things like BPIN, which can be used in palladium cross-coupling reactions, and also ortho substitution, which is desirable because these tend to be hard substrates to use in cross-coupling reactions. And then also we were able to see a bunch of hetero-aryl halides work in the reaction, which is also really desirable for metachemists because these multi-nitrogenous arene rings are really commonly found in drug molecules. I think the most important part of the reaction for me was to look at the activated alkyl halide scope, and we were really happy to find that a wide variety of different types of alkyl halides were excellent substrates. So esters were excellent substrates, as shown here, as well as alpha chloroketones, as shown here, so both cyclic and acyclic. And finally, amides, alpha chloramides are also excellent substrates, including amides that have free NH bonds that typically can't be tolerated using enolate chemistry. So these were all very exciting results to see. And then, as I mentioned earlier, you can also generate these benzylic radicals through halogen atom abstraction, and although you can't do them through enolate chemistry, so we were really excited to find that you can actually do this in our reaction. And then finally, we looked at these free carboxylic acids, which I mentioned earlier aren't tolerated using traditional palladium enolate chemistry. So through an in situ TMS masking protocol, which is basically you generate TMS protected acid in the vial, we're able to couple these in the alpha arylation reaction in similar yields as the esters, with just using an aqueous workup to give us quantitative access to the free acid product. So you can imagine that these carboxylic acids are these are good functional handles. In organic chemistry, you can use these in decarboxylation reactions to do coupling to generate radical disposition on single electron oxidation and decarboxylation. So our group has developed a variety of photoredox mediated, photoredox catalyzed reactions that allow us to do decarboxylative functionalization. So using a protocol developed by our group, we're able to do this formation of this kind of diaryl benzene motif in good yields. And also in a reaction using proto-decarboxylation of this product, we're able to generate this aryl CF2H product, which is known to be a good bioisosteer, which means that it looks electronically similar as a lot of other motifs, and we're able to access these divergent benzoic functionalities in just 2 steps from very easily attainable, commercially available starting materials. Okay, so the question, the fact that the natural question to ask in this alpha arylation method is, can we extend this method toward an anti-selective alpha arylation? These optically active alpha arylated motifs are found commonly in natural products and drug compounds. But it's a really classical challenge because once you arylate this product, you decrease the pK of this alpha proton. And as a result, this usually limits enolate arylation to tertiary substrates forming quaternary centers. This is a very classic problem in a lot of enolate chemistry, enolate alpha arylation chemistry. However, when we used our method, which utilizes this mild 2,6-lutidine base, we're really happy to find that these mild conditions enable this enantioconvergent arylation using this class of ligands called bioxyligands. So these are just preliminary results, but these preliminary results tell us that this is a feasible transformation using just chiral ligands in this reaction. So this was an exciting find, and this is something that I worked on for a bit in the lab, and hopefully somebody will pick it up in the course of in the group. So with that, I just want to end by saying that none of this would have been possible without a really fantastic group of mentors in the Hauk group and in the TOS group, as I mentioned earlier. I just want to acknowledge Professor Hauk, Cindy, my first mentor. This is Dr. Colin Lamb and Professor Stephen Lopez and Dr. Shay Patel, who are all great mentors in this group. And of course, Professor Dean Tost and Mark Levin, who's also my first mentor in the TOS group. And then also, finally, I want to acknowledge Dave and Macmillan Group and for being just fantastic group of people to work with, and Dave being a fantastic mentor, and Princeton for just for having all the resources and all of these administrative support to allow us to succeed here. A lot of funding from different types of funding sources for our group. And with that, I'm happy to answer any questions you have about anything I just talked about.
Thank you so much, Tiffany. Can everyone please join me in giving Tiffany a virtual round of applause? Just as a reminder, for those in the Zoom audience, if you would like to ask a question, you can either raise your virtual hand by clicking on the participants window, or you can type your question in the chat and I'll read it for you. For those joining us from YouTube, you can submit questions by sending an email to the email that's listed in the description box below the stream. And that email is steminar.questions@gmail.com. And I will read your questions for you. To start, I have a question for Tiffany. So I was wondering how often you use the skills that you gained in your humanities classes in your research.
That's a great question. And I think that one of the biggest things words of advice I got from mentors was to just read a lot in chemistry. So on a daily basis, you want to try to read, you just want to read a lot of papers. I think that by the biggest thing I gained from just humanities was being, writing essays was a big part of it. And then reading to write those essays required you to kind of parse a lot of really dense text in a really straightforward way. And that allows you to kind of, that part was really helpful in kind of tackling, especially denser mechanistic papers in chemistry. So that part was pretty critical to helping me learn how to read chemistry papers. Yeah.
Awesome, thank you so much. Looks like we have a question from YouTube. They say, great talk. Did you have to know how to do the computational chemistry before you did it?
That's a great question. I absolutely did not know anything about computational chemistry when I went in. I was completely very, very, very fresh. So I very, very new to this. I think think that it's really important to kind of just keep an open mind when you go into a new lab, in the sense that wherever you go, wherever you start, you're going to start with kind of very if you have no background, then it's still okay, because people are there to help you. And there are a lot of resources for you to kind of, they'll guide you to the resources that you need. So for me, when I started, basically Cindy and all the mentors in the HAL group, they just, they gave me a lot of literature to read to understand the background. We want to start with some textbooks and things like that. And it was a steep learning curve. And as of course, it's very frustrating in the very beginning, because there's a lot you don't understand, but a lot of it is that you will learn as you go as well. So not only do you learn just by reading, but as you learn, as you do your research, you'll learn a lot just from you hit the boundaries of your knowledge, and then you have to read about how do you solve a certain problem you run into. So yeah, so I guess to answer your question, I didn't know anything about computational chemistry, and it was really hard to it didn't really know anything about it, but you quickly learn by choosing good mentors and by reading a lot.
That's awesome advice. This next question, also from YouTube, is kind of related. Do you still use computational chemistry in your current research?
Yeah, I guess I do. I think it's kind of a different skill set, because it's a different type of computational chemistry in the sense that, not different type, but I definitely do a lot more radical chemistry now. So I've been trying to look at some, right now, some metal organometallic reactions, and photoreactor reactions are quite different. There's something called time-dependent DFT, which you can use for reactions. And so it's kind of, so the computational work I did in the HAL group really laid the foundation for me to be able to understand what DFT is capable of doing, and it's been useful. I've also used it just for fun for projects. We have molecule of the month meetings in our lab. I'm kind of the person. I'm a person who can model a lot of these synthetic steps for these types of meetings. So it's coming. It's good. It's a great tool to have in your toolbox for if you want to be an organic chemist doing some synthesis, or yeah, it's a really useful thing to have in your back pocket.
Yeah, that's so cool. Are there any questions from the Zoom audience? Roberto, I see your hands raised.
Hey, thanks, Emily. Thanks, Tiffany, for that talk. It was awesome. I had a question kind of like how Emily's was, but so I know you were in anthropology. Like, were you ever interested or exposed to natural product chemistry? I feel like in anthropology, and having gone where you went, I feel like a lot of people are probably talking about chemicals or certain natural products in the area. Was that ever something that you were ever interested in?
Yeah, actually, that was kind of my first interest. I kind of stumbled upon there's a lot of like, especially like in like South America, there's a lot of isolated natural products there, and they're used in, like, the bioactive compounds are used in like kind of medical applications. And so I was actually really interested in total synthesis for a little bit in the very beginning, before I realized. So I guess the funny thing is, like, I thought the Hout group did total synthesis for a bit, because they did in the past. But so that was kind of one reason why I applied for the Hout group, because I thought that I'd be able to get my hands wet in synthetic chemistry. But it actually, at the point when I did apply, they had kind of phased that out. So it just was kind of like a kind of a happy accident that I did wind up like really liking computational chemistry regardless. But that was actually one of my first interests, is like total synthesis. So that was a good a good hypothesis from you, that, yeah. But yeah, so but that kind of like goes to show that you, I think I was really lucky in the sense that if you keep an open mind, like, I didn't necessarily get the position I wanted. I didn't get a total synthesis position, but even if you don't get the thing that you want, you can still kind of it still will help you in the future to, like, if you want, it'll definitely help you in your career to learn new things. So yeah.
Thanks.
Yeah. Are there any other questions from the Zoom audience? If not, I have a question from YouTube. So they ask, both of these great presentations have radicals. How do you know you need a halogen to start the alkyl radical? Why don't these conditions work with CH activation?
That's a great question. So I guess there's two main components I guess I'll address here. The first thing is, so there's something called, so I mentioned briefly about polarity matching. Basically, you want to have to make it a kinetically facile abstraction. What you want to have is a radical that's like a nucleophilic radical abstract from something that's a more electrophilic like partner. So that if the polarity is mismatched, so in the case of a silicon center radical and like a CH bond, which is also slightly more nucleophilic, that won't be kinetically facile. So that's like the first part, the electronic part. First electronic part. The second part would also just be a thermodynamic part, which is if you have a silicon, so the silicon chloride bond dissociation energy is like 113. That's pretty strong. I think the silicon hydride and the silicon hydrogen and the carbon hydrogen bonds are not quite so thermodynamically favored in terms of the differences. So those are the two main reasons why that CH abstraction wouldn't necessarily work, and wouldn't necessarily, that's not what we observe in the system. Yeah.
Awesome, thank you so much. Are there any more questions from the Zoom audience? If not, thank you everyone for joining us for our third and final seminar. This seminar series would not have been possible if it wasn't for James Oakley, who helped us recruit all these amazing speakers, and for Dr. Ife, who helped us organize these talks. He is our club advisor. I'd also like to thank the Princeton University Chemical Society for collaborating with us on this seminar series. And finally, I would like to thank all of the speakers that volunteered their time to speak with us. Thank you so much to Marissa, Nick, Beryl, Kieran, Patrick, and Tiffany. And with that, that concludes our seminar series, but not for the Zoom audience.